In this video, I will demonstrate the sliding filament model and how it outlines sarcomere shortening and thus muscle contraction. A sarcomere is the functional unit of muscle contraction. Here, we see a relaxed or at rest sarcomere. This is the same sarcomere in a contracted state. Many sarcomeres contracting as this one is dictate muscle contraction in an organism. The following slides outline the process of muscle contraction on a cellular level. Before muscle contraction is possible, two things must be present: ATP and calcium. Muscle cells are comprised of two main proteins and several accessory proteins. The main proteins are myosin, shown here as the orange structure, and actin, shown as the round pink winding structure. In a muscle cell deprived of calcium, a filamentous protein called tropomyosin is wound around actin and covers its myosin-binding sites. Tropomyosin is shown here as the blue rope-like structure. Calcium is released from the sarcoplasmic reticulum through voltage-gated channels when an action potential reaches a muscle cell. Calcium ions have been depicted as red circles. The presence of calcium causes a conformational change in another protein, this one a globular protein called troponin. Troponin is comprised of three subunits, one of which has a receptor to receive calcium. Once calcium is received by troponin, troponin changes its shape. The shape change in troponin moves tropomyosin away from the myosin-binding sites on actin. Therefore, calcium is a necessary precursor to muscle contraction. Once troponin moves away from actin's myosin-binding sites, myosin is able to bind to actin. When myosin is bound to actin, the whole complex is referred to as actinomyosin. Once myosin and actin have bound, ATP moves in and binds to the myosin head. This separates actin and myosin. Myosin then hydrolyzes the ATP into ADP, an inorganic phosphate, symbolized by Pi. Myosin and actin reattach, but this time myosin attaches to a different binding site on actin. This distance is fixed, 36 nanometers, and is referred to as unitary displacement. Myosin releases Pi and performs a power stroke. The actin then moves. After the power stroke is completed, ADP is released. Relaxation is regulated by the sarcoplasmic reticulum, which stores calcium, and parvalbumin, which is a calcium buffer in the cytosol of the muscle cell. When all calcium is removed, troponin moves back to its original conformation, which causes tropomyosin to recover actin's myosin-binding sites. This way, actin and myosin cannot be bound and the muscle is relaxed. If both calcium and ATP are available, muscle contractions may be initiated. The force of a contraction is proportional to the frequency of action potentials reaching the muscle cell. The more action potentials, the more force of contraction. Work, by definition, is force times distance. Thus, every time a twitch, or a muscle contraction, takes place, work is being performed because a force, which is myosin's effect on actin, is being applied over a distance, which is the unitary displacement, or 36 nanometers. Finally, demonstrating muscle contraction on the macro scale is Ernie,